the overall topic that I chose to do for my presentation was the biomedical and health sciences category, and the title for my presentation and project was Crosstalk between protein mislocalization and apoptosis resistance, which was the implications for cancer progression and neurodegenerative pathogenesis. To start off, I just want to go over what I will be covering in this presentation today. I will be going over a brief introduction and background, then I will move on to the methods along with the results, and I'll have a discussion over all the findings and a conclusion along with next steps, and leave the floor for any questions that y'all might have. So for starters, the aim of my research project was to find a link between common pathways in apoptosis and protein localization, and the goal was to be able to utilize these findings to develop treatments targeting protein pathways seen in neurodegenerative and cancerous diseases. So to start off with the background, I do want to go over two key terms that I will be using frequently in this presentation. The first one on the left is apoptosis, which is also known as programmed cell death. It is an essential function for maintaining homeostasis by removing harmful cells and also making sure that there isn't too many cells in the body that might be able to form tumors or diseases that are caused by excessive cell growth. And on the right, we have protein localization, which is mainly just the process of correctly placing proteins in organelles within the cell so that way they function properly. So these two processes, even though they have totally different functions, they both aid in the formation of cancer and neurodegenerative diseases. But even though these two diseases are such big issues in society, there isn't currently much information explaining how these processes interact with each other to form these diseases, which prompted me to research on this topic. Now, moving straight into my methodology, we started with a comprehensive literature review for about 40 research articles and research papers, and with that information we got from the literature review, we were able to identify key proteins which are listed on the right, and we were able to utilize those proteins to develop 6 protein-to-protein key interaction networks that were constructed by the string database. And the reason that we used this approach was to conduct a computational analysis and to aid in visualizing the relationships involved in apoptosis, protein localization, and proteostasis, just to see how we could base our treatments based on what we could see through the visuals made by the string database. So now for our results, on the left we have figure 1, which was made by the string database as I mentioned before. This is a protein-to-protein interaction network of apoptosis and protein localization that is centered around T53. And this protein-to-protein interaction network showcases the associations among TP53, MUC1, BCL2, CASP3, CASP9, NONO, SPFQ, PRKACA, and HSPA1A. And the thickness or the amount of lines seen between interactions shows the confidence level of the interactions. So as we can see on the top right, we have 5 key interactions that we deemed as high confidence, meaning that they were in the range of 0.7 to 0.9 and above. And for example, we can see BCL2 and TP53. Over here, that connection over here has about 5 lines that are connecting it, which indicates that it has a higher thickness, meaning that we can classify it as high confidence as well. And these findings that we found with this figure confirm that the proteins associated with apoptosis regulation, stress adaptation, and nuclear RNA processing are all part of an interconnected network, which supports our hypothesis that mislocalization and dysfunction of these proteins contribute to resistance and proteostasis failure in cancer and neurodegeneration. The various high confidence scores in multiple key nodes on the string model, as seen on figure 1, showcases and strengthens the relevance of these interactions. And additionally, it also brings attention to the potential targets for therapeutic treatments. So now moving straight into figure 2, we have an apoptosis specific network which was created with the T53, BCL2, BAX, MCL1, CASP3, CASP8, CASP9, FAS, FADD, and APAF1 proteins. And on the right, as I said earlier, we have our 5 key protein-to-protein interactions with the highest confidence level out of the entire figure. And these findings help us confirm that the proteins that are associated with apoptosis inducement, regulation, and termination are all intertwined together, supporting the hypothesis that a lack of these protein interactions can lead to an abundance of cells or mutated cells that don't die off, and it could eventually lead to the formation of cancer. The numerous high confidence scores with multiple key noteins such as T53, CASP9, and BCL2, which are the most prominent in apoptosis regulation functions, shown on the string model in Figure 2, help strengthen the relevance of these interactions in terms of cell growth. Furthermore, it also highlights areas of focus for future treatments for cell growth diseases such as cancer, as we can target these apoptosis-specific pathways to help control the irregular cell growth. Now for Figure 3, we have a network that showcases a protein mislocalization network, which was formed with the proteins such as MUC1, TP53, BRCA1, PRKACA, EGFR, PTEN, AKT1, and CTNNB1. And on the top right, we have our main protein-protein interactions that were the most prominent in this figure. And these findings help confirm that the mislocalization of proteins can lead to a disruption in key pathways that regulate apoptosis and cell survival. Because of the incorrect location of these proteins, it can affect the functions of tumor suppressors such as TP53 and PTN, which further contributes to tumor development. It helps support the hypothesis that the mislocation of proteins can lead to incorrect functions being expressed, and it can ultimately lead to the formation of cancerous diseases, as I said earlier. The disruption of protein locations facilitates an abnormal amount of cell growth to occur, which leads to cancerous diseases, as I said previously in my other slides. The numerous high confidence scores with multiple key proteins such as T53, BRCA1, and EGFR shown on the string model on the left strengthens the relevance of these interactions in terms of keeping cell growth in check, and brings to attention that when these central proteins are incorrectly placed or don't function correctly, it creates issues for numerous other proteins that aid in regulating cell growth, as these proteins are the most interconnected with the other proteins shown in the model. Additionally, these key interactions give researchers treatment targets for combating cancerous diseases. And by targeting these interactions and proteins, it could pave way for aiding to control unwanted cell growth when the proteins are misfunctioning. So in Figure 4 on the left, we have a protein-to-protein interaction network of neurodegeneration and protein aggregation network. And this was created using the proteins NONO, SFPQ, SNCA, PARC7, LRRK2, HSPA1A, and UCHL1. And these findings help suggest that the placement of proteins is crucial to maintaining neuronal functions and preventing the formation of neurodegenerative diseases. This supports the hypothesis that mislocation of these proteins, especially the ones that are highly involved in RNA and splicing and DNA repairs, such as the NONO and SFPQ interaction, which is seen as one of our higher level interactions as it has a confidence score of 0.9, can help jumpstart neurodegeneration. The multiple high confidence scores with the self-contained proteins in this network, shown as displayed in Figure 1, highlights the need for these proteins to work correctly, otherwise all the other proteins would not function as well as they need to and form somewhat of a domino effect. The interactions between NONO and SFPQ is crucial to preventing neurodegenerative diseases from forming, as they handle the main processes of maintaining neuronal health in cells. This analysis has provided key areas that can be further studied to find treatments to diseases that are due to neuronal death, such as Parkinson's disease or Alzheimer's. In Figure 5, we have an interaction network of autophagy and apoptosis crosstalk network. This is created with the proteins BECN1, ATG5, ATG7, BCL2, Cas3, mTOR, and ULK1. And these findings can help imply that autophagy and apoptosis are necessary for maintaining cellular homeostasis, and they're key for preventing diseases such as cancer and neurodegenerative diseases. And making sure that there is a balance between these two functions aids in keeping the correct cells alive and the mutated ones destroyed. It supports the hypothesis that autophagy and apoptosis regulation intertwine with each other and play roles in preventing the formation of cancer and neurodegeneration. The multiple high confidence scores with the central proteins, which include BECM, ATG7 or ATG5, and ULK1 in this network, show that without even one of these interactions, there can be too much neuronal damage or too much cell growth caused. So it highlights the importance of making sure that there is a balance. The specific protein interactions, for example, ULK1 and mTOR, which is one of our higher confidence level interactions, as I listed earlier, make sure that there's a balance between apoptosis and autophagy functions, and all protein interactions in this network are crucial for cell health. And now for our last figure, figure 6, we have a protein-to-protein interaction network of a tumor microenvironment in apoptosis-resistant network. And we utilize the proteins FGFR1, FGFR2, FGF2, VEGFA, HIF1A, TP53, BCL2, and MCL1. And these findings help demonstrate that the growth factor signaling in hypoxia response can determine how apoptosis is inhibited and how it leads to the formation of cancerous diseases. It supports the hypothesis that the environmental conditions can also determine how a cell lives. And the loss of key proteins such as TP53 can lead to apoptotic resistance to form because TP53 is such a big apoptosis regulator protein. And this makes therapies ineffective in treating cancer. The mutated cancer cells can bypass normal procedures for cancer cells such as FGF2 and the FGFR1 and FGFR2 pathway and promote its growth to help them spread. The findings give us valuable information on how these disabled pathways can be targeted to prevent the abundant growth of these cells and help stop the eventual formation of tumorigenesis. And now, from all these findings that we found from the literature review and the computational analysis, it helps us suggest that the interactions with the proteins NONO, SPFQ, HSPA1A, TP53, MCL1, and BCL2 are potential proteins and potential pathways that can be targeted when treating cancers and neurodegenerative diseases, as these are the most prominent proteins. And we also noticed that specifically TP53 and BCL2 malfunctions were the proteins that were most commonly observed in the literature review and computational analysis, highlighting their importance in majority of the interaction networks we previously saw in all 6 protein-to-protein interaction networks. And however, we do want to highlight that the use of technology throughout the study does limit the reliability of the results, and we do suggest that future studies should opt for a scientific experiment for more accurate results on these proteins. And for our conclusion, we were able to identify the key interactions and proteins that help formulate these 2 types of diseases, which are TP53 and BCL2 interaction network, which aids in responding to cellular stress and controlling apoptosis. We have the NONO and SFPQ interaction, which aids in RNA splicing and DNA repair. And we have the HSPA1A protein, which refolds proteins when they are misfolded. We have the Cas3 protein, which mediates apoptosis, and the Cas9 protein, which regulates the mitochondrial functions. And for our next steps, we do suggest, once again, like I said earlier, that a scientific experiment that should be conducted rather than relying on computer models, as it can have more accurate data. And we should also suggest that future research should attempt to connect oncology with neurology to improve current or develop new therapeutic strategies that can target these common pathways in cells seen in both diseases. Thank you so much for listening. Overall, I would say I had a pretty good experience because going into this program, I wasn't sure how to start with research. I was interested in it, but I didn't have any mentors or guidance in where to start. But the program, at least, and being able to be partnered with the one-on-one mentor helped me figure out which idea, like, the idea I wanted and helped me go into it very specifically. So it helped me achieve my goal and all. So I am actually wanting to pursue medicine, and I really want to go into the research side of it. So I believe that this project would help me already gain like prior research experience. So now maybe when I go into a lab or start researching under a professor in college, I would have some prior experience to know what to do. It will give me a good baseline.